It really is my pleasure today, as the moderator, to welcome you all here at the International ALS-MND meeting, Ask the Experts. We're delighted that many of you can be here in person today, but we're also thrilled to be able to have everyone participate globally through webcast. And as a reminder to those who are listening in, you will have an opportunity for Q&A and questions and discussions at the end of the session. So I'm going to start out with an overview of things, but it really is a real honor to be doing this together with a distinguished panel, Dr. Amar Al-Chalabi, Dr. Michael Benneter, and Dr. Rick Bedlack, who really are going to give you very different components of the challenges and the opportunities we have towards treatments for ALS. And Dr. Amar Al-Chalabi is going to really give us some views in terms of genetics, and certainly anything that we don't capture in the presentations, we will be delighted to answer any questions in the session afterwards. Dr. Benneter will address areas of drug development, biomarkers, and we then move to Dr. Bedlack, who has really been pivotal in the field with ALS Untangled, and will give you some insights of approaches to moving therapies forward from his perspectives. So I hope that in the next couple of hours you will feel that you've had a lot of your questions answered, or if it raises new questions, we look forward to the continued dialogue. And I just wanted to get my first slide up, please. So again, we are tasked to try and give you as much in a very short period of time, and my goal is to give you a little bit of a sense of what is happening at the ALS Association. But before doing that, and I really am going to do this very briefly, I want to give a few key points about the disease that puts it in context of what our challenges and our opportunities are. I want to talk a little bit about our strategies at the ALS Association, talk very briefly about treatment development as an introduction to our panelists, and also then highlight some excitement around precision medicine and personalized therapies. So I use this slide often, and there are many different versions of this, but it's only to remind us, none of you in the room or those of you listening in are unfamiliar with ALS, and in some way you are affected either personally or a family member. But what we have to remember is the complexity of what ALS. You understand your disease and how you're living with it, but from the biology side, this is a very complicated disease, and the upper motor neurons, the lower motor neurons are making exquisite connections in a very precise way to be able to make the movements and the breathing that we require. And so it's all of these components, as well as the cells involved in the disease, that are key to what is going on. And although we call it a motor neuron disease, and we focus a lot on what happens with these nerve cells that connect from brain to spinal cord to muscle, it's really the surrounding milieu that's incredibly important. And so to your left, and I'm just getting used to where I have all my, here, to the left we have the motor neuron, which is in itself a complex nerve cell with many branches that are connecting exquisitely. You have the astrocytes that are important as support cells, and then the motor neuron, the nerve ending itself, has a very complicated connection with the muscle. So when we're thinking about the disease, we have to also think about the complexity of the biology that we're trying to address. Not only that, many of you are well aware that ALS is not only a motor neuron disease, but is often in combination with frontotemporal lobe dementia. And this, unlike Alzheimer's disease, a little different, is certainly a key component that we have to think about in terms of both living with the disease, managing the disease, and how we think of therapy development. And so I'll leave it to my experts on the panel to go into this further, but just recognizing that the causes of ALS, although we know some clear mechanisms and causes involved, many of them are a very complicated combination of the environment and genetics, making the picture even more complicated from a treatment perspective. And so at the association, just for ease of discussion, really, we try and think of the various areas that we support and fund globally in these different components, from biomarkers, disease model development to clinical studies. And central to this is really a person living with the disease. And in fact, our strategies over the years have included the global efforts and a translational program to bring treatments to people with the disease, living with it now and with the hope that they'll be meaningful for those who in the future. We really feel strongly that this is a partnership, it's a team effort, both with scientists, clinicians, and funding organizations, as well as the academic and industry sector. We also feel that by supporting the structure for clinical centers, clinical trials, we can more rapidly expedite clinical trials. And certainly, I'm talking a little bit more here of a US perspective in terms of clinical trials, but I know organizations like the MNDA are very pivotal in bringing the networks together, and groups globally, Leonard Vandenberg amongst others, are very involved in making sure that centers of excellence are central to our treatment development. Building consortium efforts is a clear goal because, as I said, it's going to take people globally to make the difference. And then finally, although it's easy for us to think of all the brilliant ideas, and we have colleagues to advise us, it surprises me that doesn't surprise me that there are fantastic ideas out there, and we try very hard to strategically fund as well as solicit ideas globally. And importantly, we try and leverage our investments with partnerships to really make the investments we have go further. And it's in an audience that is so familiar with this, needless to say that I do think, like we might say, genes have changed how we look at ALS. I think the Ice Bucket, the ALS Ice Bucket Challenge, has changed the world of ALS, and in some ways brought us together globally, because people living with the disease now know that globally people are understanding the disease. They understand ALS and not only motor neuron disease. And so for us, it's been great in terms of really bringing teams together and, of course, increasing funding. And so in thinking about how do we wisely use Ice Bucket money, and many of us, and I don't stand here alone to say this is how one might reflect, in thinking carefully of that strategy, we think about then, why is it that so many treatments have failed? So many of you are frustrated to hear of potentially this treatment, and then a trial goes on. Biogen is the most recent big example where they had the dexpramipexole trial, and that failed. Why do they fail? And so, is it the target that is actually the challenge? Are we getting the right target? Is the drug treatment approach correct? Can we actually measure the effect once we have a trial and we say, well, is that we can't see a real change in survival, but maybe there's an effect on an individual person? Has the disease been diagnosed too late? Is this really not just one disease, but a very heterogeneous population? And of course, is one treatment sufficient? Or for a complex disorder like this, do you need multiple treatments? And of course, all of the above are things we need to consider. And that's why in thinking of how do we spend, we were very fortunate to receive 115 million, a good proportion of that going to research, but it takes 1 to 2 billion to develop a treatment for a disease with many, many failures, not special to ALS, not true only for ALS, but true of all diseases, in particular diseases of the brain, really, really challenging. So here at the early stage of the pipeline, can we really make an impact by better understanding what it is in the pathway of the disease that we're treating through biomarker development, gene discovery, disease modeling? Can we improve? We hear the story over and over again, well, the mouse is getting better and the humans are not. I question whether the mice were getting better in some of those studies. A mouse is not a human, but they are very, very valuable tools. So how can we be more clever about using our tools and relating that more closely to the human disease to have more chances of success in the clinic? And then can we improve our clinical trials? Absolutely. Can we think of more creative designs? And can we be sure when we've had a clinical trial that's failed, we know that it failed because the drug was not appropriate, it didn't get to the target, and it didn't impact that mechanism that we know is important. And then finally, an effort going on in the US, but certainly also has been done in Europe, and the EMA has put together guidelines. Can we help build a guidance together with the global community to better educate and move treatments into clinical trials and through clinical trials more expeditiously? And so what we know is it's a very promising research field, and we'll hear more about genetics. It's the right time for exciting things to happen. We know a very complex picture of the disease, as I illustrated, with many things going on, but these are all the targets that we can think about treating and developing. We know in familial ALS we have at least over 60% of the genes connected to familial ALS, and the C9 mutation, a very, very big part of that, so extremely exciting. And with all of that in mind, we thought the first big impact things that we could do, and no, we didn't conjure them up overnight. We had been building these plans, and we had business plans together, and we didn't have the funds to do them. We had 4 major initiatives, but central to this was people with ALS had to be a big part of that, and they are indeed involved in every single one of these components through providing tissue, providing clinical information, and providing samples. And so through these efforts, we feel that we can get at least closer to identifying better targets, defining the population of ALS better, and hopefully then, although precision medicine is a complicated one because we hope we don't get such small subgroups that eventually we have a single treatment for a single person, we hope we can identify groups of people in doing so. And so I think I'm going to get the 5-minute warning very soon, and I've got lots more to say, but if I only get through this is to say that I think we can, as a team, make a huge impact. The New York Genome Center is physically located in New York, but is really working globally with programs to help the sequencing, the data analysis, and so anyone interested as a scientist to participate, this is a growing commitment. It was set up for all neurodegenerative diseases, but we were able to, with our investment, make a mark and have an ALS-focused center of that. Project Mine is a global initiative of which the ALS Association is funding, through Ice Bucket funding, the US arm of Project Mine. And I want to give credit due here to Bernard Muller and the people living with disease that had this brilliant idea, and knowing that technology is advancing and the costs are changing, that every person with ALS should have their sequence. And it doesn't mean that immediately you'll have an answer to ALS, but with all that data, we can then start to understand better what is different with someone living with the disease compared to someone who doesn't. This is driven in the US by John Landers and Jonathan Glass, but there are many others, and many are coming together here to discuss the global effort. I touched on the fact that the mouse is not everything, and yes, we have the Mouse Bucket Challenge, but in reality is, together with many other model systems, we can really start to better understand these systems in conjunction with the disease. We have a dog that has ALS and carries the SOD mutation, for example, and we're doing a lot of the pre-screening in those dog models. We also need to use the imaging opportunities and also the cells from patients that can then put the picture together. For this reason, and together with Steve Finkbeiner and others, we built the consortium in California, and we also have a core now in California led by Clive Svensson, and working globally with a team to help standardize protocols. We have many other centers that are involved in developing IPS lines. This is just one that we are significantly supporting, and we brought all those centers together to give us more of a sense of how to more quickly develop motor neurons in culture and standardize it so that when one industry is using it for a drug screen, this could be comparable to other work being done elsewhere. ALS ACT is a partnership with the ALS Finding a Cure Foundation and GE, who is very focused on imaging in ALS, and so we have many components to that program. And then just after the Ice Bucket, to be able to solicit more broadly, we set into place many programs. We received over 700 letters of intent, 230 full applications. These are in different categories listed here. And importantly, we had more than 50 specialists involved in the review, and we've issued 70 new awards in all of these different areas. And so, again, it's because of the partnerships we've been able to leverage so many different things. And I did want to, before I get to the concluding remarks, just thank all of those involved in advising and doing the reviews. It certainly was a very much a collective effort. So a few remarks on developing treatments before I lead into my colleagues. One of them is the impact that organizations like ourselves can make, in fact. And so this is an example and really exciting. It shows what we can do involved in the nonprofit area to push drug development forward. So in 2004, we funded an academic group working with ISIS Pharmaceuticals to develop an antisense technology. This technology had never been used for a neurological disorder. And in fact, we didn't have programs at that time that could fund that kind of an initiative, because in fact, the initial cost came at $400,000. Then the primate study came out at $750,000. Amounts that were at that time well beyond what we had initially put into our programs. But through a partnership with the industry and other funding organizations, the MDA in the US and NIH, we were able to de-risk this opportunity such that, in fact, now this antisense technology will shortly be in trial for a second time, but now completely taken up by the cost of it by a partnership with Biogen, IDEC, and ISIS. Very simply put, it is a way to shut the production of a protein that you don't want. And in fact, what is so encouraging is that you can actually then put this in the trial. It's put in intrathecally in the space around the spinal cord, and it gets to all the cells that are affected in the disease. This was the pivotal experiment that Don Cleveland, Richard Smith, and then later Tim Miller did to show that this was a technology that was feasible for neurodegenerative diseases, and in fact now is being used in Alzheimer's and Huntington's, in spinal muscular atrophy. So an impact in the ALS field has broad-reaching impact. And what is more exciting is that now the C9 mutation, only discovered very recently in the last few years, is using that same technology and will be in close follow-up in trial for patients with a C9 mutation. And very simply, what we're shutting down here is that orange triangle you see in the image, a very exaggerated large expansion that should not be there and is there in patients. So what we have been able to do with that first seeding opportunity is to really escalate what we can do in this program. And we now have 9 new contracts through Ice Bucket funding this year. One of them is focused on developing a treatment for C9, but a different approach to the antisense, and this is in partnership with a Swiss company, Neuroimmune. The other is 2 gene therapy approaches, one with Treeway, the founder of Treeway, Bernard Muller, who founded Project Mine, and Voyager Therapeutics, a genetics company in the US, in Massachusetts. A very interesting scientific phenomenon of nuclear trafficking. We'll hear a lot more about at the symposiums coming up. And a company who has cancer compounds is looking to see if this might be effective. TDP-43 efforts, this is also funded by the NIH with Aquina Pharmaceuticals. And then new delivery systems, GSK, and a small molecule inhibitor of C9. I'm going to close with, I hope I've got 2 more minutes to just very briefly tell you about personalized medicine. And not that we're going to give every single person a different medication, but to recognize that there might be clusters or groups of it. And this is one project done by NASM and GE in a partnership through the ALS Finding a Cure Foundation under our program called ALS ACT. The idea is that you will, in a time frame in this particular project of 12 months, capture 50 people that that don't have ALS, 50 that do. Look at various components, their imaging of the brain, look at a tracer form of inflammation in the brain, collect DNA samples, do all the genetic sequencing, and look over time to see if this changes and to see if you can then group things together. I don't have time to go into this, but alongside that, there is a big initiative called GTEC. This is a partnership with Biogen and the New York Genome Center. And there's a big, I'm going to click through these, and they will be available. These are the participating centers in that big program. And we have a big program that we are partnering with, spearheaded by Team Gleason, and Steve Gleason in particular, called Answer ALS. So we have huge opportunity moving forward. And finally, I wanted to acknowledge we have to invest as well in the people that do this work. And it's a real thrill. Many of the photos here are of young fellows several years ago who now have their independent laboratories. But I think the teams of people that we can as organizations and in partnerships put together and the work going on puts together a very promising landscape for ALS research. And on that note, I'm going to turn over to my colleague, Dr. Amal Al-Chalabi, who will give you more insights into what causes ALS and the genetics.
Thank you, Lucy, for that very nice summary. I'm going to talk about what causes ALS and try and put things into perspective. The reason it matters to know what causes ALS is because if we're trying to design a cure, we need to understand the nature of the disease to make a rational design of new drugs so that they can be targeted correctly. This is my head reconstructed in 3D from MRI scans. And one of the key problems in ALS is understanding why this particular set of neurons, just here in the motor cortex, and down here in the bulbar region of the brainstem and also in the spinal cord, why the neurodegeneration is restricted to those areas. Why doesn't it affect all nerves? This is a key fundamental problem in ALS. The advancer isn't working.
Yes, it does eventually.
Does it? Okay. That's it. Thank you. So this is a description of what happens to all of us. We're conceived, we're born, at some point we develop a severe illness, in this case a motor neuron disease, ALS, and then that disease carries us off. The causes of that final illness act somewhere. Thank you. The causes of that final illness act somewhere between conception and the onset of that illness. And there could be one cause, multiple causes, or they could be acting at different times, or maybe acting as triggers to some underlying cause. But we don't meet people at that point. We meet them after diagnosis. And so we're trying to find out what causes the condition, the things that have happened before diagnosis, from people after they're diagnosed. And that poses particular problems. So how can we understand the causes of ALS? Well, one way is to think of it as we think of most conditions, really as a combination of genes, environment, and some other things that we don't fully understand, which we can call random effects, because they'll appear to have randomly caused the condition. And of those three things, it's very difficult to understand random effects with science. Environmental studies are very difficult because we're meeting people after diagnosis. And the only way you can really get at environmental studies is through questionnaires at the moment. And trying to ask somebody about something that may have happened in their very early life, for example, how well did you sleep when you were 3 years old? Or did you used to eat apple cores as well as the whole apple or just the outside of the apple? There's millions and millions of things you could ask, and most of them are impossible to recall. So tackling environmental problems is actually very challenging. There are ways around that, which I can discuss later. Parental genes, understanding the genetic causes of the condition, is a tractable problem. It's technologically difficult and it's very expensive, but it is something that is at least a finite search space that could have an answer. So that's where we tend to focus our resources. This is one way you can think of how ALS happens, that we're born with some kind of genetic load. At some point, as time goes on, we age, we develop cell damage, and we also have environmental exposures. But unless we reach some critical burden of those disease-causing factors, we don't develop disease. So that's gene, time, and environment, those three components. For some people, they do develop enough of that burden that they will then develop, in this case, ALS, and that's, we think, a self-perpetuating process that then progresses. And in some people, it seems that the genetic load is bigger, and in other people, they may have lots of small genetic risk factors and more environmental risk. And in many people, probably, there's an underlying genetic risk, but actually, that genetic risk only matters in the particular context of the things they've been exposed to in their lives. So if you're not exposed to those things, you won't get the disease, or if you don't have that genetic component, but you are exposed to those things, you won't get ALS. So you probably need both. So how can ALS be both environmental and genetic? A big problem to understand is if you carry a disease gene, how come it only affects you when you're 50 or 60 or 70? You've had this since conception. This graph you can see here is a risk curve. It's how likely you are to develop ALS as an individual. So you've probably heard the incidence of ALS is 1 to 2 per 100,000 person years, or the prevalence is 5 per 100,000 population. Those are very difficult units for a normal person to think in. I don't think in 100,000 person years units. But if you think what's your risk as an individual of developing ALS, it's 1 in 300. That means of every 300 people you meet, one of them in their lifetime will develop ALS. So if you came over on a plane to Orlando, if it was a jumbo jet like I did, there were probably about 600 people on that plane, so 2 of those people will develop ALS in their lifetime. And you can see from this risk curve that the risk is greater in males than females, and the risk increases with age. As far as we know, it just carries on going up. So one way we could explain this is if ALS is a multi-step process. And there's some very complex maths on this, which I'm happy to go through with people individually if they want. But the bottom line is that if ALS is a multi-step process, then we could plot a log of the incidence with a log of the age. So how frequent is it at each age? And if that's a straight line, then that suggests it's a multi-step process. So we can test that with population registers, which are registers that capture every single person who develops ALS in a particular time frame in a defined geographical area. And if you do that, if it is a straight line, then the slope of that line gives you a clue as to how many steps are involved in ALS. So this is the result from the Southeast ALS Register in the UK. So you can see it looks like a nice straight line. The R-squared value there is 0.95. R-squared is a measure of how straight that line is. How straight do those dots line up? So the closer it is to 1, the better. So that's a 95%. That's extremely straight. This is the result from a Netherlands population register. R-squared is 0.99. And from an Italian register, 0.95. An Irish register, 0.99. Scottish register, 0.97. So you can see these 5 population registers, all independent. They're all European population registers, but they're all independent registers. They all suggest that ALS is a multi-step process. And what's interesting is they all give, there's the overall score, 0.99, they all give the same slope, which is about 5. And if you add 1 to that number, that gives you the number of steps that ALS is on average, how many it takes. So that's a 6-step process. So what that means is these gene, time, environment exposures that are leading you to develop the critical burden of disease, there's on average 6 steps needed to reach that threshold. And that explains a lot of things about ALS. For example, it explains that genes and environment are needed because some of the steps may be genetic, some of them may be environmental. It explains why it comes on in later life, even if someone carries an ALS mutation, because you have to have lived long enough to have been exposed to the other things that will carry out the other steps. It also explains why one gene can result in different diseases. For example, why can expansion in the C9orf72 gene cause ALS or frontotemporal dementia or both or nothing? And it's because it depends on exactly which steps you take. If you take steps A, B, and C, and D, E, F, you may have a different outcome from if you took steps Z, Y, X, and D, E, and F. It also explains why some people don't develop ALS even if they carry an ALS gene variant, because they won't have necessarily gone through all the steps. And it explains why ALS could have many causes. Of course, this assumes that ALS is one condition, and we think it may well not be one condition. But how do we find the causes of ALS? Well, it's impossible really in a relatively rare condition to do this as a single lab on your own. You have to work together. And this is something that's happening much, much more now. Mainly because of the internet and because of connectivity, and also because the ALS research community is very tightly knit. So we have international consortia. These are just 3 of the ones I'm involved in. I'm involved in many more. The Strength Consortium is one I lead. Its aim is to look for subgroups of people with ALS. So this personalized medicine that Lucy was talking about earlier. Because if we can find the subtypes of ALS, if we can show that ALS isn't really one big blob of a condition, but actually there's lots of different types, it may be that we should target clinical trials at one type, or that we should analyze our clinical trials taking those types into account, because maybe it works in one group and not in another. It'd be like if you gave penicillin to everybody with a chest infection, but only some people got better. You wouldn't necessarily know that if that subgroup that was susceptible to being cured by penicillin was only a small group. You'd have to know, okay, they've got the bacterium that's susceptible to penicillin, that's why they got better. We're also finding the genetic causes of ALS. That's very difficult on your own. Again, you have to have tens of thousands of samples because you're looking for small common variations or very rare variations that have a big effect. Finding markers for ALS also needs you to have large groups of people because we need large numbers of sample. And clinical trials require large numbers of patients to take part so that we can understand the outcome. So with all of this, we get better understanding and therefore a better chance of making a proper treatment. So how can we think of ALS in different ways? How can we work out what these different groups are? So one way is to think like a neurologist. When I see people in clinic, I say, well, this person has typical ALS. This person had a floppy weakness affecting both arms. It's quite symmetrical. I'm going to call that flail arm syndrome. This person has swallowing difficulty. I'm going to call that bulbar onset ALS. Each neurologist has their own particular classification. This one's mine. And what you can see on these graphs is what's called a Kaplan-Meier curve. And that's a curve of how long people live with the condition. The way the curve works is everybody starts off alive, so that's at the 1 level. It's the proportion of people left alive as time goes on the x-axis. So that means that the more horizontal the line, or the more to the right the line is, the better the survival. And my classification is the one on the left where it says neurologists, classification predict survival. So I just took the information I had available to me in clinic on the very first day I saw somebody, and then many years later we went back and looked at the survival of those people. And you can see a proportion of people actually live far more than 10 years. But for most people, the situation is not that optimistic. You can also see that some groups of people do quite well. So that purple line, which is the flail leg syndrome, people with that condition have a 50-50 chance of surviving about 6 or 7 years. Now, we gave the same information to a computer and said, can you classify, using this same information but with no clues, can you classify this information into groups? And it came out with 5 clusters. And then we looked at the survival, which we hadn't given the computer. We looked at the survival of those 5 clusters. And what you can see is the computer pulled out from information at the first visit this yellow horizontal line. That's 3 or 4 people who didn't die during many, many years, during 20 or 30 years. It also pulled out this group with the reddish line, which is people with an average survival of about 15 years. So to the computer, it was obvious that there were these subgroups that have much better survival, but these aren't apparent to a neurologist. So understanding what are those subgroups is very important. This is an example of a genetic study. This is a comparison of 17 million genetic variants in 13,000 people with ALS and 25,000 controls. So at each of those 17 million genetic variants, we've asked the question, does that variant occur more frequently in the people with ALS than the controls? And then we've plotted a dot on this graph. So 17 million dots on this graph. Every time it goes above this very faint horizontal dotted line here, that means there is a significant difference between the two groups. And you can see this large skyscraper in the middle. This is called a Manhattan plot because it looks like skyscrapers. There's not much of a Manhattan skyline at the moment for ALS. But what you can see here is this is the C9orf72 gene signal. But you can see there's actually about 8 signals there. So we can use that to try and dissect out what's the jigsaw puzzle, what are the proteins, what's the pathway that's causing ALS. We're also taking part in Project MIND, funded by the Motor Neurone Disease Association in the UK. That is an international whole genome sequencing study aiming to sequence 22,500 people's genomes. To put that in context, to sequence one person's genome cost $3 billion and took 16 years the very first time it was done. It now can be done in about a week for about $1,000. So the technology has really rapidly advanced. And the main problem actually is handling the data. Each person's data comes back as 1 terabyte of data. So we're going to end up with 22,500 hard drives before they're even backed up, before they're plugged into computers and analyzed. It's actually the data analysis that's the challenge here. But what's very good about this data is it has multiple layers of information. We have the clinical pattern for that particular person. We often have other information that went with that person. They may have filled out an environmental questionnaire, or they may have also given us a sample for what's called epigenetic studies or for RNA studies. So we have multiple levels of scientific information on each sample. And genetic discovery is doubling every 4 years. So what we're hoping is, by understanding the genetic risks, we can then put together the jigsaw puzzle and understand what causes ALS. That doesn't mean that all ALS is genetic. What it means is there is a genetic contribution. And that means that, just as I showed with the multi-step model, you need more than the genes, you need the environment as well. So just to conclude, international collaborations are vital for us to make progress. Technology is only part of the answer. We have to have patient participation, and we also have to have creative ways of answering the scientific questions, such as using computers or using clues from the cancer world like we did the multi-step model. Thank you very much.
Great. So that's a real good segue into some of the issues that I'd like to talk to you about. And you'll hear some common themes that are emerging from what Lucy's had to say and from what Amara's had to say. But what I would like to do is try to take a little bit of a deeper dive into some questions around why is it that we've been so singularly unsuccessful in developing therapies for this disease. And it's not for lack of trying. Here's sort of a sample of the drugs that have been tested in the last 20 years. So there have been dozens and dozens of clinical trials in phase 2, in phase 3, trying to determine if there's a treatment effect. And with the one exception of riluzole, which is really exerting a very modest effect, we've really not been successful. And so it's worthwhile to step back and to ask, why is it that we have failed in the way that we have? And what can we learn from the experience that we have so that we can move forward and bring therapies to patients with this disease more efficiently and more expeditiously? So what I'd like to do is I'm going to run through, and I'll just bring these up. I only have 20 minutes, but I want to talk about 8 challenges and 8 opportunities that each of these present. And there's some language here that I'll try to explain. You've been introduced to some of it already. The first point is that there are many different causes of this disease, number 1. Number 2 is, irrespective of the cause, there are many different biological processes going on, many things in the nervous system that are sort of part of the biology, the disease process, and these may be different in different people. Lucy has touched on the limitations of the animal models that we have. I'm going to say a word about the outcomes that we use to measure whether a drug is having an effect. They have serious limitations too. I'm going to talk about what we don't really have a lot of. We don't have good biomarkers or biological markers of the disease, and I'll try to explain what those are. Dr. Al-Chalabi's touched on this and shown you some graphs, I'll show you some others, that this disease is actually quite varied from one person to the next, and that's a challenge in terms of determining whether an experimental drug is having an effect. Dr. Bedlack will talk much about this, so I'll be very brief, that clinical trials and research can only move forward with patient participation in research programs. And then lastly, an idea that we've been thinking about and working on for many years is the question of whether we're bringing treatments to bear too late in the course of disease. So here's a model. Look at the bottom part of this figure first. So here's time, and here's some measure, just arbitrarily, maybe not arbitrarily, of motor neuron number, motor neuron function as the principal nerve type that's affected in this disease. And we think perhaps as we age, there's some loss of motor neurons in all of us. At some point, the disease process begins. But that doesn't appear, it doesn't manifest immediately. It's like when we get an infection, there's an incubation period. You're infected, but you don't have any signs or symptoms of the disease yet. That progresses for some period of time, during which time it's pre-symptomatic. Symptoms then appear. It then on average takes about a year before people get a diagnosis. Some further months go by before somebody may enroll in a clinical trial. And then the disease runs its full course. And so one concern, and I'll come to this with the last challenge, is whether we're bringing therapies to bear just too late in the course of the disease. So to give you a sense, when we study cause, we have to study cause before the disease begins, not just before symptom onset. You have to study it before the whole disease process begins. So when does it begin? Number 2, the biological processes we can study throughout the course of the disease. Here's number 3, the animal models, the preclinical models. They don't fit so well into this schema. Here's number 4, outcome measures. How do we measure whether a drug is having an effect? We can only measure that in people once they've already developed symptoms, right? Otherwise, there's nothing to measure. That's where biomarkers come in. These are things that we can measure whether people are showing signs of disease, whether people are not yet showing signs, whether they might even be at genetic risk, but otherwise quite healthy. So this is a major sort of challenge. Number 6 is this variability of how the disease appears in different people. Here's research participation, involvement of people with this disease, people who may be at risk, family members in the disease, and this notion of intervening early, that we should be intervening when disease begins, maybe before, very soon after symptom onset, but not way out here. So let's sort of talk about each of these in a little bit of detail. So here's the etiology, the cause, okay? And so the idea here is that there are many different causes. And so Dr. Al-Shalabi gave you the example, if you're treating a chest infection with an antibiotic, there are many different infections that don't all respond to the same antibiotic. Another example might be if you have anemia, a low blood count, that might be because you're vitamin B12 deficient, might be because you're iron deficient. And if you treat everybody with anemia with iron, only those people who are iron deficient can have a hope of responding. So we need to understand the cause if we're going to then think rationally about how we develop therapies and then give those therapies to people who can actually benefit from that treatment. So we really need to understand cause to be sure that we're not giving people inappropriate therapies, that we're bringing the right therapy to the right sorts of people. You've seen this figure before. This is the growth in the genetic causes that have been identified. We now have 25, 30 different genetic causes among 15% of people. So there's still amongst 85% of people with this disease, we don't know the cause. So if there are 30 causes amongst 15%, how many more causes or components or contributors to cause might there be that we're yet to discover? And that's a huge obstacle. That's a huge challenge in terms of thinking really rationally about therapy development. But one opportunity that immediately presents itself, and Lucy touched on this, is to think about clinical trials in subsets of people where we know the cause, and in a single cause or a group of causes that are related. So you understand the cause, you have a therapy that targets that cause, for example, an antisense oligonucleotide against a specific genetic mutation or genetic form of the disease, and do a clinical trial in that subset of patients. So perhaps therapies need to be more targeted, maybe not precision in the sense of a different treatment for each individual person, but different treatments for different subgroups of people with the disease. So here's challenge number 2, the sort of variability of the biological processes that are going on. And Lucy showed you a slide of many different systems that are affected, many biological processes. And the challenge here is we often select drugs based on our understanding of a biological mechanism and a drug that then targets that mechanism. But different biological processes may be ongoing in different people. If we just take the 30 or so genetic causes that we know and we try to bin them into different biological mechanisms, there are some groups. So there is convergence amongst certain genetic etiologies or causes onto common biological pathways. And so one idea might be that we don't need therapies that are only for people with a particular genetic abnormality, but people who share a common biology. Of course, the challenge then is to understand and know that biology in life and to be able then to bring a therapy to bear, but that's another therapeutic approach. And so clinical trials in subgroups of people who share a common biology, even if not a common cause. So the third challenge is the limitations of our available animal models. What you may or may not know is that a common way in which drugs are sort of brought to clinical trials is they're first tested in preclinical models, in models that are not human. Those could be an animal, those could be a cell culture in the laboratory, but we typically need a way to identify a drug and to get a sense that this might be worth giving to people. And this is typically done in an animal model, at least until now. And so the workhorse of this has been the SOD1 mouse. SOD1 was the first identified genetic cause of this disease, and the first animal model was developed based on introducing this genetic change into a mouse. And the challenge is that we've cured ALS in the mouse, in the SOD1 mouse, many times, but this is not translated into effective therapies for people. And that's not to say that the mouse has no value, it does, but it can't stand alone. And so what we need are additional models that can tell us about other forms of the disease that are not necessarily related to the SOD1 mutation, but also we can get a greater confidence if an experimental drug is effective in more than one model. So targeting the drug to the right model, showing that that drug works in many different models, all of those can give us a much greater degree of confidence that when we bring that drug into a clinical trial, that will then be helpful for people with the disease. So I'm going to skip over some of the bullets here because I think I have too much information. So I think the opportunity here is to develop new model systems, and that's being done. There are new animal models, new mouse models that are being developed based on the new genetic etiologies that are being identified. But there's also a lot of work that's being done, and Lucy referred to this too, to generate what are known as induced pluripotent stem cells or induced motor neurons. These are cells in addition in the laboratory that can be made to behave and in fact are motor neurons. And these then become model systems in the laboratory for drug discovery and defining drugs and that might then be worthwhile bringing forward into clinical trials. So the opportunity here is to develop and to work with these new models. The fourth challenge is this limitation of our existing clinical outcome measures. And the measures that we most commonly use in a phase 2 or a phase 3 clinical trial are things like survival. What proportion of people are still living with the disease after a certain period of time? Or we use this measure of function called the ALS Functional Rating Scale, the ALS-FRS, or we use a measure of breathing like the vital capacity. These are the common outcome measures that are used, and then we compare in a group of people who got the drug and a group of people who didn't. If we look at those outcomes, which group fares better? And the challenge, and here's just a sample of drugs that have been tried, is look at the numbers of people that are needed to be in a clinical trial and look at the duration of time that people need to be followed for us to learn whether a drug at a single dose is having an effect. And so this is a great challenge. It means that we can't take promising compounds, test them at different doses in a speedy fashion to find the drugs that hold the most promise to then bring to a larger patient population. So what we need is outcome measures that we can measure change over a shorter period of time in a smaller number of people to get a sense, is this showing promise? Should we continue to study this, or should we set this drug aside and move on to the next one? So the impact on drug development of not having these sorts of outcome measures is that we can't quantify therapeutic effect over the short term. Trials are long and expensive. We don't know how to find the right dose, and it's very, very challenging to know which drugs do we bring into these big multi-center international trials of hundreds and hundreds of people that last for years. So the opportunity here is to develop novel outcome measures that are sensitive to change over the short term. Easier said than done, but that's a good segue into challenge number 5, which is the paucity of these biomarkers, these biological measures of disease. So a good example would be, say, in the field of AIDS and HIV, we can measure the viral load in the blood. We can actually measure how much of the virus is floating around in your blood. And then when you give a drug, you can measure whether you can lower that viral load, and that tells you that you're impacting something very relevant to the biology of the disease and gives you a strong reason to keep pursuing that drug. We need those sorts of biological markers in ALS, and they're beginning to emerge. We have a bunch of candidates. Electrophysiological, measuring nerve and muscle electrical activity and function, using imaging to look at the brain and the spinal cord, and then biochemical measures in the blood, in the spinal fluid, in the urine. And there are lots of promising candidates that are now beginning to move into validation stages that if we can validate those may be really instrumental in making our clinical trial process that much more efficient and effective. So I think one opportunity here is a focused attention on validating those lead candidates that we have. There's been a lot of discovery. Discovery needs to continue, but we need to take those lead candidates and validate them, show that they're really, really good and helpful, or we need to show that they're not useful, set them aside, and move on to the next set of candidates. So challenge number 6 is the variability amongst people. And although there's sort of an average survival with this disease of 3 to 5 years, as Dr. Al-Chalabi's explained, 10% of people are still living with the disease at 10 years, a smaller proportion at 20 years or 30 years. And if you put into the same clinical trial somebody who's going to live for 6 months and somebody who's going to live for 30 years, you can't tell whether that longer survival is an effect of the drug or just because somebody had a different form of the disease that gave them a longer survival. So you've seen this kind of figure, survival and the rate with which function changes vary tremendously from one person to the next. And so what we need to do is we need to characterize this, we need to define it and understand it, we need to understand what the basis for it is. Biomarkers can help here potentially in terms of predicting who's going to live for 6 months, who's going to live for 20 or 30 years, that we could then put those people into different clinical trials or subsets within a clinical trial that we're looking at the drug and a placebo in people with a disease associated with longer survival and a disease associated with shorter survival. But we need ways to predict that. And then one possibility is to limit trials or to restrict trials to people with particular forms of the disease. So I've told you we could stratify and we could do clinical trials based on a genetic cause, based on a shared biology, or perhaps based on a shared anticipation of a shared sort of future or prognosis of what that disease holds. So I think that the opportunity here is clinical trials in people whom we expect to phenotypically. What that means is how does the disease appear, how does it manifest in people who have a common form of the disease from that perspective. So challenge number 7, and I won't say much about this because I'm sure Dr. Badlack will, is that without patient participation, all these sorts of studies can't move forward. Yes, there are lots of studies that are done in cell culture, in the laboratory, in animals, but the pivotal work of understanding the genetics, understanding the environmental factors, developing biomarkers, doing clinical trials, all of this really absolutely depends upon people with the disease participating and giving of themselves to participate and to help advance science. So I'm going to skip over this next slide because I don't want to upend Dr. Badlack, who I'm sure will focus on this a lot more. But I think the opportunities here are partnerships with our patient groups like the ALS Association, the MND, the MDA. These are essential to our success in moving forward. I think we need to increase referrals to multidisciplinary clinics. That's where a lot of the research is taking place. We really need to integrate research into that multidisciplinary clinic setting so that clinical care and research become part of the same endeavor. We need to really educate investigators and physicians about research, get them fired up and enthusiastic. We need to make sure that we remove barriers to patient participation, whether those are financial, whether those are physical, to enable patients with a disease that causes functional disability and physical disability to participate. And one of the things we're interested in is remote assessments, moving assessments in clinical studies into the home to make this easier for people to participate. So the last challenge I want to touch on is this notion that we may be intervening in the course of the disease too late. And I showed you this model that I put up before about us losing motor neurons as we age, the disease beginning, some pre-symptomatic period, symptoms appearing, disease progressing, and diagnosis. And one of the things we don't know is how long is this pre-symptomatic period? Is that days, weeks, months, years, decades? We don't know yet, but it's an important issue to study. It's important not just because if we want to understand cause, we've got to understand it before disease begins. But if we want to understand the environmental exposures, you've got to ask people, what were you exposed to before the disease began? So no good taking people who are diagnosed and saying, what were you exposed to 6 months ago if the disease began 2 years ago? Right? If we want to know what environmental things contribute to triggering or causing the disease, we have to know when the disease actually begins. So we really have to understand what's going on, what's this natural history, what component of the iceberg lies beneath the water, when does it begin, and how do we begin to study that? We know from other neurodegenerative diseases, we know from Alzheimer's, we know from Parkinson's, we know from Huntington's disease, that in some of these diseases, like Huntington's, the disease may begin 10, 20, 30 years before the first symptom ever appears. Now that may not be as long as in ALS. Huntington's, after all, is a disease that runs for about 20 or 30 years, typically once symptoms appear. That's not routinely true of ALS. So the duration of that pre-symptomatic period may not be as long, but I would wager that it is a period of at least months to years. That's an opportunity to understand the disease, to measure it, to quantify it, and most importantly, to think about intervening early. So I think the opportunity here is to study disease before it ever appears, and I'll be happy in question time to address how we might do that, and an opportunity even to think about early intervention or even prevention in people who are at genetic risk for the disease. So what I'd like to then do is sort of briefly wrap up with some conclusions and just tell you about an initiative that's going on that you've not yet heard about that begins to address some of these challenges. So I think to sort of wrap up, I'd say that therapy development for this disease is immensely challenging, and I think you all have a sense of that. But I think new opportunities are emerging from insights into the cause of the disease, insight into the biology of the disease. I think biomarkers, certain types of them at least, are absolutely essential, and I have no doubt that you'll hear much more about that during the course of this meeting. I think there's a huge amount that we can learn from studying our past experience, and I think we absolutely have to, and we are as a field and as a community, because we can't continue with the business as usual of, the next drug that somebody thinks about that might target a certain mechanism and bring that into a clinical trial without the biomarkers, without understanding the biology, without understanding the differences between patients with this, with different forms of this disease. So I was going to say a word about this thing called the CREATE Consortium. This stands for Clinical Research in ALS and Related Disorders for Therapy Development. CREATE's much easier. That was a big mouthful. And CREATE forms part of an initiative from the NIH called the Rare Diseases Network. The NIH understands that people who are affected by a rare disease, and ALS is a rare disease, often there isn't the same investment from a financial perspective in terms of research to understand that disease and to develop therapies. And so about 12 or 15 years ago, the NIH established this network to study a group of rare diseases, not only ALS, not only neurological diseases. And about a year ago, we were fortunate to be able to get ALS incorporated into this network. And the CREATE Consortium is the consortium across the US, but also extending into Europe and Canada, that's studying ALS and related diseases as part of this consortium, as part of this network. And this network collectively is studying about 200 or 300 different rare diseases. And the hope is that we can learn from each other, that many rare diseases may face similar challenges, and that we can learn and help each other as we try, each within our own fields, to advance science and to develop therapies for patients with these different diseases. So this is the CREATE Consortium's network, a website. You can look it up, RDCRN, Rare Disease Clinical Research Network.org /CREATE. We have a section for patients and friends. We have a section for clinical investigators. And there's several sort of key things that CREATE is undertaking. The one is a large longitudinal study that's trying to understand this variability in phenotype and to understand the genetic contribution to that and to develop biomarkers. So trying to address three of those challenges that I spoke about. As part of that effort to develop biomarkers, we have a pilot project program where in partnership with the ALS Association we can fund pilot projects to do discovery or to do validation, and we can provide from the repository of biological samples that are collected from these people who are participating, we can provide de-identified samples to help fuel that engine of discovery. We have a training program because we are focused not just on education of patients, the community, society, but also future clinician scientists and investigators. And we have this thing called CREATE Connect, which is a form of a contact registry. It's an ability for people to sign up online, express an interest to learn about research opportunities, and to engage in a discussion and a dialogue with investigators, with scientists. And this is being done in partnership and collaboration with Dr. Bedlack. And one of the initiatives being done under this is a virtual journal club, where we're asking people who've joined this to tell us what are some of the science you've heard about in the news, online, that you would like to hear from the author of that paper that can explain that in a way that makes sense to a lay audience. So that's one of the initiatives that's being undertaken through this thing called CREATE Connect. And I've left down here an email contact address and some website information if you would like more information about that. And so I haven't seen a hand yet unless I've missed it. I missed it. I apologize. So I hope I haven't gone over my extra 5 minutes. See, it helps not to look where the hand is coming from. Then you can just keep talking. And so perhaps I'll stop there. Thank you for your attention and look forward to some discussion after our next speaker.
I'd like to thank the organizers for inviting me to talk about my projects. And also thank God that none of the other co-moderators wore the same jacket. I always hate when that happens. So I'm going to talk about two strange projects that I've been working on. One called ALS Untangled that I've been working on for about 7 years, and one called ALS Reversals that I just started. So many of you have probably heard about ALS Untangled. We started it back in 2009. within something called the ALS Research Group, the ALSRG. And we realized at that time that patients were very often going online and looking at some of the alternative therapies that were there, that were making extraordinary claims for benefit in ALS. Unfortunately, there are no internet police, so people can say whatever they want to about something they're selling on the internet and nobody holds them accountable. So our goal for this group was to develop a team of clinicians and scientists to systematically assess these things called alternative therapies and ultimately help patients and families make more informed decisions about them. We think of ourselves kind of like the X-Files, and we've got the same motto. We think the truth is out there, but it's not always easy to find. So there's 3 parts to what we do. First, where do we get our ideas? Second, how do we actually do these reviews? And then finally, how do we get the word out to folks? So we wanted to make it as easy as possible for patients and families to get ideas to us. And so we've got a lot of ways. We've got a website. We've got our emails on there. People ask me about these things in clinic. Most of our ideas come to us from patients and families via Twitter. So many of you, I think, probably know what Twitter is, but one of the nice things about Twitter is you don't have to remember anyone's email address. You can just send a tweet out into cyberspace that says, hey, ALS Untangled, I'm wondering about this clinic in Moscow that's offering electric shock therapy for ALS. And every night I mine the world for tweets that contain the word ALS Untangled. And if I see alternative therapies in there that people ask about, I put them on our website. Another nice thing about Twitter is you can get an idea of how many people are interested in what you're doing by looking at the number of people that are following you. And so I was very ecstatic this year when we crossed the 2,000 followers mark. I went home and bragged to my wife about this, and she reminded me that Lady Gaga has 33 million Twitter followers. But to be honest, she does have her own stylist, and I have to put these looks together myself. So You can also get an idea of how popular you are by how many tweets you see appearing with your name in them. And as of about 2012, we're averaging about a tweet a day, which is more than I ever thought we'd have. And as a result of all that Twitter activity, the list of things that people have asked us about has just exploded. In fact, it's now approaching 200 things that people have asked us to research. We've got so many things that we'll probably never get to all these in my lifetime. And as a result, I've put a little vote button next to each one of these things on our website. So if you have something on there that you've asked us about and you want us to look at it, be sure to go on the website and click the vote button. You don't have to pay to vote. You don't have to register to vote. All you got to do is push the button and it moves up on the list. And we've also refined the way that we do these evaluations over time. So we've always had a team. The team has now grown. There's 100 investigators now like me from across 10 countries that are participating in these reviews. We've got a real standardized operating procedure by which we go about gathering the information to construct one of these reviews. And just this past year, we actually created something called the Table of Evidence that I'm very proud of, because I don't think anyone's ever done anything quite like this before. So every time we review an alternative therapy, we ask about 5 categories. What's the mechanistic plausibility? Is there any plausible way that this could help somebody with ALS? What sort of preclinical data are there? Is there any sort of preclinical animal model that this thing has been used in, whether a cell culture model or an animal model? What do the case reports look like? Are there lots of them? Do we actually have the medical records on these people to validate that they have ALS and validate what they're saying? Are there any trials? And then finally, what do we think about the risks of this particular thing? And in each of these categories, we give grades, letter grades, ranging all the way from A to F. And then there's also a category U. And so there are very specific things that you have to achieve to move up to the next letter grade. So for example, you'd get a mechanistic plausibility grade of F if what you're saying has never been demonstrated, if we'd have to rewrite the physics books to actually explain what it is that you say you can do. For example, energy healing. So the other nice thing about a table of evidence is that it allows people that care about different types of evidence to grade these alternative therapies for themselves. So rather than have me tell you which one I think is the best, you can say, well, which category is most important to you? For a lot of my patients, risks is the most important category. So they might go on our website and they might decide for themselves which of these things I might try based on a risk grade of A. Other people with ALS don't care that much about risks. What impresses them the most is what do the case reports look like. They want to try something that other people have tried and had some sort of validated improvement on. So they might look for things that have a cases grade of A. So in 7 years now, we've completed 33 of these reviews. It doesn't sound like a lot, but they take about 40 hours to do. And we were averaging about 3 or 4 of these a year up until the past year when we've got some new support from the ALS Association that's really allowed us to kind of explode. We've doubled the number of outputs that we've had this year. So this year we've reviewed the Precision Stem Cell Clinic, we've reviewed acupuncture, a supplement called MitoQ, methylcobalamin, which is a form of injectable B12, something called Protandim, the gluten-free diet, and then most recently something from France called Endotherapia. If you want to read about these things, go onto our website and just click the name of the thing you want to read about, and it will open up a free article. That's what our outputs look like. Our outputs are actually articles that have a very structured format. They're short. We try to write them in language that non-scientists can understand, and they actually get submitted to the ALS journal, the main journal in our field, where the editor reviews them. They're peer-reviewed. They get published in the journal. So, unfortunately, the majority of the 33 things that we've looked at over the past 7 years do not appear to be very promising. And rather than take you through each of those individually, I would like to just tell you about some red flags, some things that these more bogus therapies appear to have in common. And that way, if you see something new and it's going to be a few years before we get to it, you can ask yourself if any of these red flags appear. First of all, something that's advertised as being dramatically effective against 40 of the world's worst diseases, Alzheimer's, Parkinson's, MS, brain tumors, ALS, fibromyalgia, diabetic neuropathy. You have to ask yourself, if this person really has something that works against all those diseases, why are they practicing in a small clinic next to a Kentucky Fried Chicken in a strip mall? They should be the most famous, wealthy person in the history of medicine. Sometimes things are just too good to be true. Anytime something's advertised as perfectly safe, that's a problem. Nothing's perfectly safe. People have side effects from placebos. Anytime there's a non-MD who's on a website making medical recommendations, that's a problem. Anytime you go someplace and they ask you to try something without any sort of informed consent, so you don't have it in writing exactly what it is that you're being asked to do, that's a problem. Anytime someone wants you to pay a whole bunch of money up front before you really know what you're getting into, that's a problem. And then the one that I think is the most reliable of all the red flags, anytime you hear about a treatment that somebody wants to sell you, and then they never want to measure anything afterward, that's a problem. Because anybody who's legitimate wants to know what happened to you after you try this thing. They don't want to just send you on your way after they collect your money. But surprisingly, some of the things that we have looked at over the past 7 years appear quite promising and might actually work. And so one of those is this thing called Lunasin. Lunasin is a soy peptide. It's taken orally, and it's got a lot of mechanisms, at least in cell cultures, that seem like they would be useful in ALS. Antioxidants, anti-inflammatory mechanisms, and alteration of something called histones. All of these are mechanisms that we've pursued in ALS trials before, so they're not completely crazy. So the mechanism grade is a C. What's the evidence that this might work? Well, there are no preclinical studies, there are no trials. But there is a pretty amazing case report out there. It's this gentleman right here. His name's on the internet, Mike McDuff. He's been in the news. He's reported that he has ALS, been diagnosed by multiple different doctors, and that a year before he started taking this stuff, he couldn't speak or swallow, and a year later, his speech and swallowing have recovered back to normal. So we found Mike McDuff, and we got his medical records, and he's right. He does appear to have ALS. He has seen some good people. He's had all the right tests. He's had all the right tests for mimics that I can think of. I have records from his ALS clinic, and his speech, swallowing, and muscle strength are all dramatically improved over the past year, which is not supposed to happen in ALS. This seems to be reasonably safe, but it is quite expensive. And so for the past couple of years since I saw this, I've been asking myself, what do I do about things like this? And now with the Ice Bucket Challenge, I finally have some resources, and I've decided to create a program to study people like Mike McDuff, people that I call ALS reversals. Now, it's important to realize that there may be multiple explanations for why somebody like this would get better. First of all, even though I have his records, he could have been misdiagnosed. There could be an ALS mimicker out there that even the best of us don't know about. Second, maybe there's something special about Mike McDuff that made him resistant to ALS, and whether he took Lunocin or not, he was going to get better. And then finally, maybe the Lunocin actually worked. And so in my new program, there's going to be two arms. One arm is called ROAR, Replication of ALS Reversals, and one arm is called STAR, Study of ALS Reversals. So the ROAR program is basically a series of small pilot trials using the exact same treatments that people who had ALS took them and got better. We're going to do the exact same thing they did. Now, sometimes people their eyes get wide when I mention this, but this, patient drug discovery is not new. There are many examples. So one of the most famous examples is Viagra. And people may not realize this, but Viagra was actually under study for heart disease. And it was only because patients, men in the study, at the end of the study came forward and said, I don't know what it's doing for my heart, but I'd like to keep taking this. That the company kind of said, what in the world is going on here? Patients discovered what that drug is currently used for, and now it's a $1 billion drug. And closer to home, Nuedexta, same situation, was being studied to see if it slowed ALS progression, and it didn't. But patients came forward and said, hey, I noticed something that this drug is doing to me that I like. It's helping me with that pseudobulbar affect, and now it's FDA approved for that. So, of course, it's a long shot that one of these types of trials is going to find something useful. So, to maximize the usefulness of this program, we're going to add something to it. We're going to incorporate some novel patient-centric design features to try to target increasing enrollment and retention. And we're going to test these design features as well. So Michael was hinting at this a couple minutes ago. One of the most perplexing things to me in 15 years and 2,000 patients worth of experience is that less than 10% of people with ALS enroll in clinical trials. It's pretty shocking. So why is that? Well, there's a whole bunch of reasons, which I'm not going to go into all of them today, but I will tell you about 4 of them that I've heard over and over again. First of all, people tell me we can't find a trial that we qualify for. All of your trials have very narrow exclusion criteria. You have to have so much ALS, but not too much, and you can't have this attached to you or that. Why do we do that? Well, we're trying to really homogenize the populations that come into these studies, because most studies are looking for very small effects in a very noisy disease. Typically, we're looking for a 10% slowing in the rate of decline in the ALS functional rating scale. It's a pretty small signal in a disease that's quite noisy. So what are we going to do different? We're looking for the biggest signal that's ever been looked for. We're looking for people to get better. And if they don't get better, we're going to consider our trial to have failed. And what that means is that we can take anybody into the study. Doesn't matter how long you've had ALS, it doesn't matter how sick you are, what you've got attached to you. Now realize, of course, that the chances of this working are quite low and that we might actually reject something that has a small positive benefit. But that's the risk we're going to take. Second thing I've heard over and over again, people with ALS, they don't like coming into trials where there's a placebo. There's no getting around a placebo when you're trying to figure out for sure that something works. But when you're doing pilot trials, the first ever attempt to see if something might work, there's no reason why you can't use matched historical controls now. We have so many databases. We can match people according to things that determine how fast they progress and get a rough idea of whether something's promising enough to take into a more traditionally designed study. Plus, if you think about it, if I do this study and 20 people have an ALS reversal like Mike McDuff, do I really need to do a phase 3 placebo-controlled trial? Probably not. Again, a real long shot, but just saying. Next, people don't like the burdens of trials. So, the trials, they typically require lots of visits, long days, lots of trips back and forth to the clinic, and you have to be in the study for a long time. And, that gets harder as a person with ALS gets sicker. So in this program, most of the visits that we ask patients to make are going to be made in their own living room through their own home computer. We're going to teach them how to use a website called Patients Like Me, and we're going to teach them how to measure things in themselves like the ALS functional rating scale, and periodically we're going to check in on them by phone to make sure that they're actually doing what we ask them to do. And then finally, one of the things I've heard over and over again from patients is how frustrating it is to come into a trial and realize you were one of the first ones in, and by the time that trial finishes enrolling all of its subjects, and by the time the last subject finishes the entire trial, and by the time the results are published, it might be a year and a half or 2 years later. That's a long time in the lifespan of a patient with ALS. So one of the nice things about this new program, since it's on patients like me, you'll be able to follow along in real time. You can check in anytime you want and see what's happening with the Duke Rohr Lunison study. So again, this is a very novel design, widely inclusive, largely virtual, single center, open label pilot study using historical controls. We're going to bring 50 people in. Why did we pick 50? It's based on a power calculation, which I won't go into. I'm not going to tell you about all these inclusion criteria, but just a quick glance at these will show you that there are no disease duration criteria. There are no criteria based on disease severity, no criteria based on what you have attached to you. Of course, you have to have ALS. Of course, you have to be able to use a computer. And you can't be on a lot of other experimental therapies or have had a bad reaction to Lunison. So this is a year-long study, but there's only 3 visits required to Duke. During those visits, we're going to confirm that you have ALS. We're going to teach you how to use patients like me, teach you how to measure your ALS FRS and your weight. We're also going to do blood tests because we want to look at a biomarker. We want to see if Lunison is actually doing anything that the company says it can do in people. So we're going to measure histone acetylation. We're going to ask you to check in once a month on patients like me, and we'll be looking over your shoulder for the first few months to make sure that you're doing it and not having any problems, and my coordinator will be calling you periodically to check. So that's the ROAR program. The other program is the STAR program, and this is an attempt to understand why these reversals might be happening. And again, the 2 hypotheses, other than the fact that it might be a treatment that's working, maybe it's an ALS mimic that we don't know about, or maybe some of these folks have something different about them that makes them resistant to ALS. So what are we going to do here? Well, first we're going to try to survey colleagues around the world and identify as many ALS reversals as we possibly can. I already have 22 in my desk. I'd like to get 100. Going to put them all in the same database for the first time, make sure that the records are compatible with an ALS diagnosis, see what these folks might share in common as far as past medical history or concomitant medications. We're going to try to get blood samples on these folks and look for antibody-mediated ALS mimics, and we're going to look at their blood for whole exome sequencing. Now again, this might sound like science fiction to you, but there's a precedent from another disease group. Within HIV, there are a group of people called elite controllers that seem to be resistant to the disease. They get infected with the virus, but they never get sick, and they never take any medicine. And for years, people just shrugged their shoulders like they did with these ALS reversals. Finally, somebody put them in the same database and did whole exome sequencing and found that a lot of these folks have the same polymorphism. It's an abnormality in a gene called CCR5. CCR5 is a protein that's in all of our cells that it turns out HIV needs to get in. We didn't know about that before we studied these people. And furthermore, Pfizer used that knowledge to develop a drug to block that receptor, which works for everybody with HIV. So maybe we can have a similar success by studying these ALS reversals. More information is available on several websites, ALS Untangled, the Duke ALS Clinic, and ALS Reversals. And I just want to thank especially patients. You're the inspiration and the driving force behind these projects, the funding sources, ALSA, the MNDA, and the Larry Vance Hughes ALS Foundation, and the smart people that I work with at Patients Like Me, Mass General Hospital, and Duke. Thank you very much.
So I want to thank my distinguished panel for tremendous presentations, really sticking to time and giving an incredible amount of information, which I'm sure has sparked many questions, and I welcome any question for any of the panelists. We'll all try, whoever's relevant, to attempt it. And I think there are microphones around, so if you can wait a moment, especially for people who are listening in on the live streaming. There's one question in the front here, please. And for those of you listening in, we are going to be answering your questions too. Just patient while we address a few from the room.
This is a question for the last presenter. One of the other obstacles that people find when looking for alternative medicines that I would like to add is language. Do you have all this information in any other languages?
It's a great point. We don't at this time. So if you have an idea for how we could do that in a cost-effective way, I would be all ears. Yeah, that's what it always comes down to. I mean, Google Translate works pretty well for these types of articles, but if you have any other ideas, I'm all open to them. Yeah.
Great.
Thank you.
Other questions? I see one here. Oh, yeah, here.
Hi. This question is for the reversal of ALS. Are you looking for patients who have completely reversed or even just improved?
Could you just repeat that question for me?
An
The
improvement versus just,
reversal
yeah.
should be complete or could be just partial improvement.
Yeah, that's a great question. So I've been trying to figure out how to operationally define an ALS reversal. Obviously Mike McDuff is an ALS reversal, right? He's so much better. So I'm actually going to be presenting in a couple of days here at this meeting an idea that I have for how to define it. And it's based on an analysis of a giant database that I did. The database is called PROACT. It's got over 8,000 patients in it. And so it looks to me like a nice cutoff point might be to define it by a 4 or more point improvement in the ALS functional rating scale that lasts at least 1 year. Now, why did I pick that? I picked that because, first of all, small little reversals that last for a short period of time are actually not that uncommon. About 50% of the people in the PROACT database had a period where they improved by a point that lasted 60 days. So we have this idea in our mind that ALS is just a straight line down when you get it. And it's not like that at all. There's periods where it's flat. There's periods where it bumps up a little. There's periods where it goes down faster. I want to pick something that's robust enough to be biologically important, but it's also got to be common enough that I can study. I can't just do a whole project based on 22 people. That's not enough people to do any kind of decent analysis. So I'm going to need probably at least 100. So I think with that definition, I might be able to get that many.
Great, thank you.
And do they have to have alternative therapy only or any kind of like stem cell?
Oh no, this is two completely different programs. So they're overlapping, but the ALS alternative program, the ALS Untangled, that's a program to help people understand alternative therapies. The reversals program is to try to understand why do people with ALS sometimes get better. So those people who had the ALS reversals that I'm looking for, I don't care what they took. It doesn't matter. I just want to see if they got better, I want to know about them.
Even with a stem cell?
Anything. Yeah, anybody out there.
One.
Send me an email. Send me an email. I'd love to hear from you.
Okay.
Yeah.
Okay.
There's a question here, I think, from our listeners.
I can start with one thing. I mean, so
Oh, I think we need to repeat the question. There are two questions here. One is, how close are we to a new treatment, and why has it taken so long before we actually have another treatment?
So I think, first of all, I'll take the second part of the question. New treatments in ALS come around all the time. It just depends how you define a new treatment. Like, I'm pretty excited about Nuedexta, first of all, for the pseudobulbar affect component of it. That's a symptom that I did not have much luck with for many years. And just recently, just last month, we learned that there's been another trial of Nuedexta that's actually shown benefit in helping people with speech and swallowing. So now I'm starting to use a lot more Nuedexta just over the past month in my clinic because the trial shows that it can help some people with their speech and swallowing. So I think new treatments are coming around all the time. It's just a matter of we don't have the disease-modifying treatment that we all want that stops or reverses ALS yet. We have symptomatic therapies right now.
Did either of you want to comment, Dr. Beneteau or Dr. Al-Chalabi?
I mean, only to add that I think this is, the issue of why we're not being more effective at developing therapies is is the issue I tried to address and sort of raise what are some of the factors that go into that. So I think it's about understanding the cause. It's about understanding the variability between people in terms of how they appear, what the biology of the disease is, having the appropriate biomarkers, intervening early in the course of the disease. So I think all of those are challenges that we're working to overcome, but until we make more progress in overcoming some of those challenges, it becomes hard to sort of really see the progress on the basis of having identified them. So I think one never knows when, science moves forward in small bits and pieces by a process of mass action. There's lots of little progress, and then suddenly things begin to fall into place, and there's perhaps unexpected progress. And so I think it's hard to predict when we'll have a new therapy or a new promising agent that will have that disease-modifying effect. But I think understanding or having a greater understanding of the reasons why we've struggled and making sure that we're paying attention to those issues, addressing them as we move forward, increase the likelihood of something in the near future.
I'd like to comment, and then I'll hand over to Dr. Al-Chalabi. I think that we can be very, very optimistic and positive. I don't think I can say to you next year there'll be something that's going to be effective, but I think we are probably better positioned than in the time that I've been involved in ALS in the last 20 years. One, there are more funds. Yes, never enough, but significantly more. I think as a community, we are really much better kernelized around this disease. We are partnering with industry in a much bigger way. I'm amazed how many companies are committed in this space, from small to large. Biogen could have walked away when Dex failed, and they haven't. Cytokinetics is really moving into a phase 3 trial for something that might have huge impact on quality of life. So I think if we're going to find something, we are going to make progress in these few years. And I feel it's really because of the team, because of the recognition globally that there are opportunities that we can all work as a team together. So I feel very, very optimistic, but that isn't an answer that it's here tomorrow. Sorry.
And I would echo everything that's been said already, but I'd also add that one of the ways we've tried to find new drugs is to test them in model mice. Those are mice that develop motor neuron disease, or ALS. And the mouse that's been used traditionally is one that has a mutation in a gene called SOD1. Now, that's the mouse that we can cure quite well with drugs that it doesn't seem to translate into people. But if we look at the pathology of people with motor neuron disease, if you look at the spinal cords of people with ALS, they don't usually have any pathology that involves SOD1. They've got pathology that involves another protein called TDP43. And about 2% of people have pathology in SOD1, and the rest have a problem with TDP43. So perhaps what we really need to do is develop a better mouse model, and if we can cure that mouse model, it might translate better into people. We've probably been looking in the wrong direction all these years. Another reason is that it's this stratifying, this precision medicine problem. We're looking at ALS as a whole, as a blob, as a single disease, but actually it's probably lots of different conditions, and we need to be a bit more nuanced about how we test our treatments. And the third thing I'd really like to say is the thing that's really surprised me, I appreciate those questions, the thing that's really surprised me is why we haven't taken Riluzole and tried to make a better version of it. Because with other drugs, with other diseases, as soon as a drug is successful, you get lots of me too drugs. If you take migraine, people, the first drug that came out was called Imogran or sumatriptan. Immediately there was rizatriptan, zolmitriptan, there were a whole lot of other triptans that were invented. Why haven't we got other Riluzoles? That's what I'd really like to see.
And I would add to that, just to along the theme of TDP43, that although we don't have the perfect models yet for TDP43, many more are emerging, and I think there is a lot of emphasis in academia and industry to find therapies to use TDP43 as a tool. And I think in combination with cell lines from patients carrying these mutations and others, I think we're going to make quite an impact. So although I think we're learning, as Dr. Benneter expressed, the challenge is we're already moving in the direction to fill those gaps and build those tools. So I think in combination with being more aware and building those resources, I'm sure we'll make a huge impact. So other questions? Are there some from the floor? There are quite a few hands, so we're still in good shape. We have quite some time still. If we can just get mics lined up, and then we'll take the floor questions and a few more from the webcam. You might want to keep your hands up if we're not finding mics for you. There you go.
It's extreme mental and physical, caused by extreme physical or mental or both stress. And all the genetic variations, and they all down to the coping mechanism. So my question is, in your clinic, how many people every month coming in with symptom onset, and what do you do about it? Any one of you.
So,
Yeah, I'm not sure. I'm not sure. What do you mean by symptom onset? Yeah.
So if person come in and say, I may have ALS,
Oh.
doctor, look at me, what do I
Yeah.
have?
Well, I mean, we probably see 3 new rule out ALS patients every week, and about 17 return patients every week. And you know, the thing about the places where we, all 3 of us practice, they're all academic centers. So usually they've already seen other doctors before they come to us.
Right.
So you know, a person who comes to see me with a diagnosis of ALS that doesn't have it is pretty rare. It happens maybe 2 or 3 times a year.
So can you describe what what you do in terms of diagnostics? Perhaps that's what you really want to understand.
No, what I mean is that when the person have extreme stress symptoms, and very often they don't get helped. They don't have rehab, they don't have psychiatric help and psychology. Basically, I'll give you some example. The kids in college, my son's college, they come to me and say, I'm stressed out, I can't sleep. Can I take a drug? I said, no, you don't do that. And get some good sleep. But they don't have anyone advise them to get good sleep. So they stress out. They tell me, my biological clock is not 24 hours, it's 36 hours. I said, no, this has to be wrong. So a lot of people are poorly advised. So that's what I'm getting at.
So your question really is, so you're really asking, are people, when they're newly diagnosed, how do we help them cope with that diagnosis?
No. When a person has symptom onset, can we do something at that point to quickly bring down their stress level by good sleeping help?
I think the problem is we see people at too late a stage to really be able to help. By the time someone comes to a doctor, a neurologist in the UK, and this is true worldwide really, about a year has passed since their first symptoms.
Right.
So it's too difficult to do that.
The key is, can we do something when they just had a symptom?
Yeah. The difficulty is knowing that those symptoms are from ALS and not from something else.
Well, it doesn't matter.
Yeah,
If somebody is stressed out, figure out how to help. That's my question.
We can talk more. You can talk more with the clinicians
sure.
afterwards. So we have a question here. Kathy, you want to take the question at the back? Kathy Collette? Yeah. Oh, you haven't got a mic. So
Okay. So my question is pretty much what she said just now. So she had undergone autologous bone marrow mononuclear cell therapy. We've had a lot of discussion today about the genetic reasons behind ALS. So I would just want to know, what are your thoughts about autologous stem cell therapy? And it surely has shown good results with her, but from the wider scientific point of view, what do you think about this? It's open for everybody, whoever would like to answer.
I can make a quick stab at that, and then I'll move it along. I mean, I think we obviously haven't spoken about stem cells on the panel, not because that is a potential opportunity for therapy. We just had to make some choices of what to discuss. That said, there are certainly various trials. I can't speak to the treatment and who's taking care in this case here. They are still experimental. It's unclear what the benefit will be. We have trials in the US. Obviously, Brainstorm is an autologous stem cell trial. I think it's unclear yet to say, and just as Dr. Bedlack addressed, it's sometimes a question of numbers, because is it in one case really the stem cells that is helping, or is it just the progression of the disease? With autologous stem cells, one idea is that you're getting some extra factors that are helping protect the nerve cells. Some of the autologous stem cell approaches are modifying those stem cells, but by before re-entering them. I think we're at a very early stage of really understanding what is going on. In the US, the neural stem trial, which we as an association and NIH helped fund a lot of the preclinical work, we were pleased to see it move to human trials very cautiously and carefully with the FDA guidance. I think we can say there's one reversal in that trial that people know about and is on our board. And we actually took the approach similarly to Dr. Bedlack. Jonathan Glass thought maybe it was the immune suppression protocol in using those stem cells, not the stem cells, and we're doing funding a small trial in that. So I think it's still very early in small groups of people to understand its impact. That said, I think it's something under careful clinical trials process, it's very valuable to explore further. So that's the start. I don't know if others want to comment.
Well, I think there's pros and cons to both approaches. I mean, so the mesenchymal stem cells, they're your own cells. So the nice thing is you don't have to be immunosuppressed. But the downside is they're your own cells. So if there's something genetically wrong with all your cells that contributed to getting ALS in the first place, you're just getting those cells back. And modifying them might help for a little while, but there might be a better way. And then you've got the flip side where you do somebody else's cells, embryonic stem cells, for example. There you've got to be immunosuppressed. So there's no perfect solution yet.
And perhaps just to add, as you know, is that a stem cell isn't a single thing. You referred specifically to autologous stem cells. So there are different types of stem cells. There are different ways in which they can be given. They can be given into the spinal fluid, into the blood, into the muscle. They can be modified before they're giving back. How many need to be given? So in the same way that we sort of don't think about a drug, we think about drug as nuanced. There are many different kinds of drugs. How do you give it? What dose do you give it? We think about stem cells in the same way, and these in different shapes and forms, routes of administration are currently under study.
Great.
Lucy, can we piggyback onto that question?
Absolutely.
Several questions around stem cells that you've addressed that virtual listeners have also asked. So someone else is asking, their daughter's having a baby in a few months, and should she have them put the umbilical cord cells in storage for stem cells? And how might this be useful to someone with ALS? This is certainly a common question we get.
We would like to tackle that.
Is there a use for umbilical?
Well, I'll just tell you what I tell my patients because I get asked this all the time. I tell them, right now I don't see anything on the pipeline where people are using umbilical stem cells. It doesn't mean it's not going to happen. So, if you can find a bank that can do it for a price that you're comfortable with, go ahead. But on the other hand, if it's a huge stretch, I think the likelihood of those becoming useful in the near future is pretty small.
So we advise people in the same way.
And so do we.
Great. Did you have any more from the web around stem cells?
You addressed those. Thank you.
Great. So Kathy, you've had the mic a while, and then in the meantime, if we can get a mic here, please.
Thank you. Kathy Collett from Indiana. For Dr. Benatar, the CREATE project. Do I understand correctly that that is a function of the NIH?
What do you mean a function?
It's sponsored by NIH.
It's primarily sponsored by NIH. There's supplemental funding from the ALS Association, potentially from the Muscular Dystrophy Association, but it's principally funded by the NIH.
And people with ALS register data in there that contributes to your understanding of the science, and then they may be informed of clinical research opportunities, right?
Well, so there are many different components to it. So there's a clinical, or there's a study protocol where people can make visits to one of the 12, 13 centers. They can undergo what we call sort of deep phenotyping to be characterized from a motor perspective, from a behavioral and cognitive perspective, given the link to frontotemporal dementia, contribute biological specimens, including DNA that goes for whole genome sequencing. And then we're trying to return some of those genetic results back based on the genes that we know are implicated in this group of diseases back to people. So that's one protocol that's ongoing. There are efforts to initiate other protocols. But another element of this is that online registry for people to indicate an interest and a willingness to learn about new research opportunities as they become available, so that we can help to connect people to studies that may be relevant to them in their area.
Understood. Thank you.
Sure.
But we have a CDC registry that we've spent a fortune on. And people with ALS go to that registry, put their information in. It's supposed to contribute to science. They're talking about spending an even bigger fortune on tissue samples. and then they may be informed of some clinical research. So,
And
do
so-
you guys talk to each other at all?
We do, absolutely. And the truth is, registry is a bad word and a bad term in this context. We've inherited it from the NIH's Rare Disease Network. It's not really a registry. And that's why we've sort of rebranded this as calling it Create Connect. What it really is a mechanism to connect scientists, physicians, and patients. And the National Registry, for example, advertised this. We sought their approval. They put it out as a mechanism that patients might be interested in, that the community might be interested in. So absolutely there's an intersection between the two, and we're not at all trying to duplicate what the National ALS Registry is trying to do.
Well, I think from a patient perspective, I know some of you people from Florida remember Chuck Hummer. He used to say, I feel like I've left tissue all over the country. I think people with ALS having to go here to put data in and go here to put data in. If you had to buy a book on Amazon that way, you'd never shop. But I think they're starting to feel like they're leaving their data
So
all
may
over
I
the
address
country.
that point? Because, I mean, I'll leave it to Dr. Bennett to add as well, because, in fact, Dr. Bennett is very pivotally involved. We recognize that it might feel that there are many things, and that's where we have initiatives that are overlapping. In reality, there's not one way of doing this, and, in fact, being global, there are many initiatives going on. And part of our role in working with clinicians like Michael and others, we're trying to make sure that these things communicate. And we've just worked together on a white paper for biomarkers that will come out with a concept of the spoken hub type of idea. Ideally, yes, maybe one world would have one thing that's centralized. On the other hand, actually, there's valuable bits to each of them. So our goal is to, as a community globally, be able to connect these things. Some things, for example, are the GUID, that's an identifier so that things are not replicated. We very carefully are involved with this, with the New York Genome Center efforts and Project Mine and others. And in reality, it's the value that each of these bring and their intersection that make it more valuable. So no, patients don't have to go to 10 different places. They will be interconnecting their data. And yet they might have wanted to go to 3 or 4 different places, and we will identify that they are interconnected. So we are working together to merge things. Everyone might have a slightly different style, but with Neurobank and initiatives that are becoming very much more interconnecting, I think we have a huge opportunity. And I do think this is not duplicating. One added to that, where we're collecting samples through New York Genome Center, Answer ALS, GTAC, and we have Project Mine. Reality is that we are going to have to validate studies. We are going to have to be able to be absolutely sure that this particular gene is linked to the disease. And one of the biggest things we all talk about as a community is having good control populations to compare with, because little changes, and I'm not the geneticist, I don't want to talk for Dr. Al-Chalabi, but little changes are in the general population. How do you compare? So I do think that these initiatives are going to intersect in a very meaningful way, such that every single study has a value. Sorry, I don't know if you want to add to that.
No, I think
But could I just add, Kathy, I think you're right. I mean, one of the things that we need to start looking at, by the way, I'm Barb Newhouse from the ALS Association. One of the things I do think we need to start looking at is being able to have a person with ALS be able to go to one place and put in information, natural history information, if they're going to give a tissue sample, give it all in one place so that we are connected in a way of doing that. And I know we're all interested in working toward that, but I don't want to lose Kathy's point. I think it is difficult. Well, I know you won't lose your point, but I don't overall want us to lose the point that it does become cumbersome for patients to have to go to several different places and put in information that is either essentially the same or similar.
So maybe I didn't say it clearly. They don't have to go to multiple places. They go to a place that is comfortable to them, near their clinic. Some things are, so the repository, the biomarker repository being set up with the CDC registry is a remote one. It's a very different concept because not everyone in the country, in the US, has access to clinics. But absolutely, and I think that Dr. Al-Chalabi should speak to how it's being done in Europe. There is no one place that everyone can go to. But if we can interconnect their data and they go to what is comfortable in their places would be helpful. So I think you could maybe comment.
Yep. So the way we do it in Europe is people go to their local centre, they donate their clinical data, they answer environmental questionnaires, They may give a tissue sample or a DNA sample. And really what you're asking about is data sharing, because the issue comes down to that sample is analysed by the relevant expert in the field. So, for example, if it needs a genetic analysis, there's only 2 labs that do that to a high level in Europe, and it'll go to one of those, and then they share the data with everybody, not only the people who donated not only the lab that donated the sample or the clinic that donated the sample, but it's available to everybody within the European consortium. And actually, we also make that data available to people in the US and Australia, provided that consent has been given for that. So I think it's a very simple system. You basically have donation at one centre, and then the data is shared as it needs to go. The sample can be sent out to where it needs to go as well for other things. So, for example, if you have a blood sample, that can be used for RNA, that'll go to the RNA centre. A bit of it will go to the DNA centre, and a bit of it will go for methylation analysis in an area that does, in a centre that does epigenetics.
And I should emphasize that principles of broad data sharing are enshrined, for example, in the consent forms that we use in Project Create and in other consortia, so that de-identified DNA sequence, biological samples can be shared as broadly as possible with whomever the expert is, wherever they are.
And another key feature, which I think actually happens in the US and in Europe, is we have a single identifier that's anonymized, that you generate the same ID from the same input information, so that we know if there are duplicate samples. So the way we do it is very similar to the way that it's done in the States. So you give the first name, the last name. We also have to account for different spellings, and we have a single alphabet that will do it. Date of birth and year of onset is what we use, and then that's used to encrypt to generate what's called a hash. And that's a very, very long string of letters and numbers. That's a unique ID for that patient. So if they go somewhere else, it'll generate the same ID, and then in the database they'll be matched up as the same person.
And can you use those data that you take from the patients in Europe for epidemiology studies too?
Yes, we do. So the data I showed in my talk, that was from 5 different population registers. Those are some of the epidemiology studies that are done. We've got tens of thousands of people in epidemiology studies.
We have another question here.
So we actually have 2 I'm going to put together from 2 different virtual listeners. The first is, does edaravone, also known as Radicut, slow the progression of disease? And another one from someone living with ALS that has taken a look at the Deanna protocol and would like some comments on that from the panel.
Sure, be happy to. So, with regard to the first one, edaravone, so this was presented at the American Academy of Neurology, but the paper, to my knowledge, has not been published yet. We've been looking for this paper, and so until the paper comes out, I'm not going to have much to say about it. That's one of the things I learned in my years at the helm of ALS Untangled, is you need the information in front of you before you really should say anything. And when it comes to the Deanna protocol, that is something that we've reviewed. And so just so people have a framework on this, this is a collection of supplements that were put together by an orthopedic surgeon whose daughter was reportedly diagnosed with ALS, and he was trying to find something to help her. And his report was that her progression slowed on this. And so when we reviewed this, of course, we reached out to him, we reached out to her, we asked for records. They did not send her records, so we can't confirm that she has ALS or that her progression has been slowed. And in fact, I'll say I've yet to find a single person that I'm convinced had their progression slowed by the Deanna protocol. It gets a mechanism that's plausible because it's a number of supplements that do different things that could be involved in the biology of ALS, but there is at this time an animal study that's highly flawed, that's never been replicated, and there really is no interpretable human data that's come out on that. So when my patients ask about it, I just tell them I don't think it's all that promising at this time. I think if there was something there, we would have seen some people that you could actually point to and say, look at the difference in their progression before and after the Deanna protocol, and we don't have that. So.
We have a question here. Oh, sorry, Nicole, was there another one?
Thank you.
Is there any research being done for familial ALS where you study people with the genetic predisposition, measuring the behavioral and environmental factors that could trigger ALS?
I think it's a Michael question.
Yeah, so we started a protocol looking at this about 7 or 8 years ago, and it's been growing slowly. And the model is to take families where the disease runs in the family, to identify the genetic cause in the affected individuals, and then to take first-degree family members, so siblings, children, and to do pre-symptomatic genetic testing, with appropriate counseling and helping people choose and decide whether that's something that they want to do, and if they do, whether they want to learn the results. So we carefully take people through that. And then we follow them longitudinally over time. So these are people who are asymptomatic, they're well, but they may carry a gene that puts them at a high risk of developing disease. And not only do we sort of look at them from all sorts of perspectives, from a motor perspective, from a cognitive and behavioral perspective, using imaging and physiology, we also collect environmental exposure data. So we collect it real time. So the first time that we meet somebody, we ask them from as far back as they can remember until now. But each year they come back to see us, we ask about what's happened in the last year. So it's sort of more contemporaneous. And the idea is that over time, unfortunately, some of those people who carry one of those genetic mutations will go on to develop ALS. And then we can go back and ask, were we able to measure things before the disease ever appeared? Was there anything about the environmental exposures that they reported that might be relevant? As you might understand, this is it's a difficult study to do. It's slow going, and you have to have a long view of this, because people who are at genetic risk are at genetic risk over their entire lives. They're not necessarily at genetic risk over the next 6 months or the next 3 years or the next 5 years, a period of time over which funding agencies often look to think about this sort of protocol. So it's a challenging undertaking, but one that we are doing and have been doing for the last 7 or 8 years.
Anna, you might want to add.
No, that's fine. Actually, I think it's covered it all.
There's one more question here.
We have a new question from a virtual listener. How important is positive attitude? And if it's important, how can we fight the doom and gloom that comes to us when we attend clinic?
I'll just say, from my perspective, positive attitude is very important. And as far as fighting the doom and gloom, I mean, the way that I do it is I try to focus on the things that I can control, not the things that I can't. So, for example, I can wear a fun jacket when it's clinic day, and I can put a smile on people's faces. But it sounds silly, but that is one small thing I can do that day. So, I mean, if I sit around and think about what I can't do, whether it's in my work with ALS or anything else in my life, I'm going to be miserable. So I just try to focus on the small things that I can do.
And I think a lot is also trying to help people focus on what they're still able to do, and what are the things over which people still have control. I smile when I think about Tony Judt, the British historian who was diagnosed with ALS and has written quite a little bit about his disease. And one of the things he said is that he focused on what he could still do. He could still write, he could still think, he could still boss people around. He writes about it humorously, but I think helping people sort of think in that framework. But I also think, and this is part of perhaps trying to integrate research into the clinical environment, is that I think there's hope through research. We've seen tremendous progress. You've heard Lucy's optimism and an optimism in the field that we're sort of standing on a threshold based on progress that's being made. And I think trying to communicate that sense of optimism, but without overstating it, I think is a real important part of what should go on in a multidisciplinary clinic environment. And I think that helps.
And I'd like to add that we've done a study in which we've examined exactly that question. And we found that having a purpose in life, which could be anything from having a job or having a child to look after, any purpose in life, has an effect about as big as the effect of riluzole.
Wow. Interesting. Are there questions on the floor? I see one here to the right. I think we have 5 more minutes to wrap up. So.
I wonder if each of you might be willing to say what you think might be the most promising line of research or lines of research to find treatment or a cure.
Starting on this side. So I'm super excited about my ALS reversals project, but to be honest, what I think is most likely to work in the next few years is the antisense oligonucleotide stuff. So the nice thing about that is that we've got a defined subtype of ALS that we're trying to treat with those, where we know exactly what caused it. We know exactly how these drugs work. And I think those might actually give us the ability to stop the progression of ALS in its tracks in those people that have those genetic mutations. So we'll see.
I would agree. I mean, I think that they present an opportunity where we really know the cause and one's able to go after the root cause of the problem. So whether it's with an antisense oligo, whether it's some other gene therapy-based approach, I think the notion of taking a subgroup of people where we know the cause, we know how the disease unfolds over time, and offering a therapy that gets at that upstream biological mechanism, I think it holds the most hope.
Yep. And I would add that it's really the fact that we now have the ability to do massive, large-scale collaborations that are truly international, tens of thousands of people participating, many centers from lots of different countries, and we can study different levels of information from the same person. That really makes a much, much more powerful data set for analysis. I think that's really going to lead us forwards.
And I would echo about the antisense, but to also really say an area that I think is advancing quite remarkably, it's a tough area, is the biomarker area. I'm quite encouraged by it. I think that there are a few, Dr. Benetton didn't go in the specifics around disease progression markers, but neurofilaments are really looking very interesting. Iron horse diagnostic is one example, but I know there's huge work going on in Europe where we're looking to validate the neurofilaments as a potential. And what is clear is that they seem to show prognostic, so how quickly the disease will progress. So talking to Dr. Bennett's idea that you might group those kinds of groups together, so another defining marker is one example. There are many others that are being tested that might be of value. A marker in the urine we'll be seeing more data of. So I'm very optimistic. It's a very difficult area. Even in Alzheimer's and other neurodegenerative diseases, we struggle with that, although there are things emerging there too that I think we have hope that that might change our clinical trials trajectory. So optimistic about that. We have to wrap one more question here and one more question there, and I think we have to wrap up then after that. So from the webcam.
Yes, so this session has been extremely informative. I'd like to share with my colleagues, and they're asking if it'll be accessible online. So I'd just like to answer that yes, we're recording this. We'll certainly post it on the ALS Association website, and we will share that with our partner organizations as well. And another person had asked if they had wanted to contact any one of our speakers, how would they do that, or will that information be something that we could share?
Can put our email addresses.
So we can provide that. So Nicole, we can work with you to provide that. And I was reminded that I think there's a watch again option on this webcam as well. So that's a way to see it all again if you wanted to follow it again. And that really will be posted for some time, at least in the next week coming up, and maybe longer. So we'll work on that. And I believe we have one more question on the floor here. The final question here.
Thank you. I'm Lori Fieldman from the Les Turner Foundation in the Chicago area, and I wanted to go back to the positive attitude question. I have the privilege of counseling patients and their families in home, so I work more with the quality of life than the quantity of life your research is trying to increase. But I was wondering, did you say that you had a study and can you say a little bit more about that in terms of purpose? Because I talk with families about that at least 2 or 3 times a week.
I'm happy to send you the paper. It was.
I would love to see that.
It was basically a qualitative study, so it was a semi-structured interview of a set of patients and carers. And it was looking at multiple different aspects, and the thing that really found out was having a purpose in life. But that could be, the purpose was undefined, so it could be anything like a hobby, a family member who cared about very strongly, etc.
Yeah, we see that all. We're fortunate enough to have a home team, and we all see that. Yeah. So I would love to see that. Thank you.
So on that note, if others in the room have questions, I know the speakers will stay a little longer to chat with you. Thank you so much for all the listeners in from all over the world. I hope that you found it valuable. As we said, the resources will be available, and we really welcome any questions about any of the studies or other things that come to mind. A special thank you to our panelists who have taken their time from very busy schedules, and in fact to researchers and clinicians globally for their hard work and commitment to this. So thank you.